Hello everyone, my name is Nikhil Mittal and I am a PhD student at Biomedical Engineering Department working in the field of mechanobiology. I am about to present my poster on Myosin-independent mechanotransmission for sensing the stiffness of extracellular matrix. Most cells in our body need to attach to a solid environment called extracellular matrix for their survival. The stiffness of this matrix is critical for the regulation of various cellular activities such as wound healing, tissue development, and even malignant cancer transformation. Cells sense the stiffness of this matrix by transmitting mechanical force through a sensor complex called integrin-based focolidation. Different forces lead to different signaling pathways and responses. Therefore, an understanding of how cells sense and respond to stiffness can help in identifying treatments for developmental disorders and cancer. This force versus stiffness relationship has been explained by a molecular clutch model in which F actins retrograde flow dynamically clutches with the cell ECM addition, resulting in the force transmission to the matrix. Non-muscle myosin, a motor protein, is dominantly involved in the force transmission as its pulling force is transmitted to the matrix as traction force. A significant number of studies have shown an increasing force trends in response to the increasing stiffness. However, these studies have primarily focused on myosin 2 mediated mechanisms for stiffness sensing. As we know that myosin contractility is one of the most common effector in response to mechanism signaling, recent studies have shown that cells are able to transmit small but significant force even in the absence of myosin activity. Therefore, it is important to identify how cells transmit differential force even without myosin to shed light on the very initial stiffness sensing event. As an alternative power source, actin polymerization by two actin nucleators APO3 and formin can contribute to the retrograde flow. Therefore, we hypothesize that different force transmission is possible independent of myosin contractility through actin polymerization-driven retrograde flow. To test this hypothesis, we use turf microscopy and elastic gel coated with fluorescent beads as an extracellular matrix. The cell deformation on the gel is quantified by comparing position of the beads before and after cells. This combination of gel assay, imaging method, and analysis is called traction force microscopy. The cell and bead images are analyzed using a MATLAB-based TFM software, where displacement field is measured using particle tracking velocimetry. The force maps are then produced by solving the inverse problem using a regularized Fourier transform traction cytometry. The level of force is quantified and compared or different stiffnesses. A control experiment was done with Paxil and mRuby expressing 3D3 fibroblast cells to corroborate with the previous study, which demonstrated an increasing trend with increasing stiffness. Next, cells were treated with a myosin inhibitor, blebbistatin. A differential force trend was observed in myosin inhibited cells, which strengthens our hypothesis. Further, we inhibited either of the two actin nucleators in addition to the myosin using SMIF-H2 for formin and CK666 for ARP3. To our surprise, a biphasic trend of force against stiffness was observed with low force at intermediate to high thickness. With this data, we speculate that formin-mediated polymerization could lead to fast actin flow, which can slip in the absence of ARP3, resulting in not enough clutching and less force transformation. In contrast, actin flow speed might be slower in ARP3 polymerization, which can be stickier, resulting in better clutching for force transmission. All this data suggests that initial ECM stiffness sensing even by focal adhesions is regulated by actin polymerization based retrograde flow. Thank you.